Welcome back to my channel. Hope you all are doing great. So now today we will learn about erythrocyte abnormalities. So there are many types of erythrocyte abnormalities. So let's get started. The first one is leptocytes. So what are leptocytes? Leptocytes means thin cells. So these are thin RBCs which appear as rings of membrane with large unstained central areas. So these can be found in the example can be severe iron deficiency, thalassemia, and liver disease. So now here is the pic showing leptocytes. In this pic you can see these RBCs are very thin and only the periphery is dark. So these all are leptocytes. Now let's move on to the second cell, which is our target cells. These cells are very important for your exams, and you will find in every second, third slide you can find target cell in the lab. So what are target cells? Target cells have the central stained area with peripheral rim of hemoglobinized cytoplasm, which means the hemoglobin moves to the peripheral of the RBCs. These cells are resulting from disproportionate large surface area to volume, which means the cells which have the RBCs which have disproportionately larger surface area as compared to their volume. Now these cells can be microcytic to macrocytic. The example of target cells in which diseases they can be seen are CLD. Let me change the color. CLD, hereditary hypobeta-lipoproteinemia, iron deficiency anemia, and some hemoglobinopathies which are specific. We can say target cell will be present in all these hemoglobinopathies like HBC, THEL, HBC, sickle cell disease, sickle cell, beta thal, or HBE. In all these hemoglobinopathies we will find our target cells. So in this pic if you can see the target cells which have centrally stained area, then pale area, then peripheral rim of stained area. These all are target cells. So the first pic here we can see is of alcoholic liver disease, and the second pic here shows the patient with HBC disease. In this also we can see target cells, numerous target cells are present. Okay, so I hope you are getting it. Let all you are clear. Now the third cell is stomatocytosis. Now the stomatocytosis meaning, the word means mouth. Now in these type of cell the central area is slit like, as you can see in the diagram, and these cells are sometimes considered as an artifact. Now example of stomatocytosis are alcoholic liver disease and myelodysplastic syndrome. The pic here shows the stomatocytosis. These are all are stomatocytes in which there is a slit like central area. Now let's move over to most important cell of the day, which is sickle cell. Now the sickle cell, by its word you can understand that it is seen in sickle cell anemia, and it is typical for HbS homozygous states patient. And one important thing finding here is that when these cells are subjected to anoxia, there is presence of massive sickling. When these cells are subjected to anoxia, these cells produce massive sickling. And other findings which we can see are target cells in sickle cell anemia, Hobel-Jolly bodies in splenic atrophy of these patients. The pic here shows the sickle cell, typical sickle cell, and these cells are boat-shaped cells. This also pic here shows the boat-shaped cells in the patient of sickle cell anemia. Okay, I hope you are getting it. So let's move on to our next topic of the day, which is erythrocyte inclusions. There are only mainly two types. First one is Hobel-Jolly body. Oval jolly bodies are nuclear remnants. Okay, so what are these? These are nuclear remnants, and these stain purple on Romanowski stains. The example of condition in which oval jolly bodies can be seen are: first one is splenectomy, the second one is splenic atrophy, third one is celiac disease, and fourth one is pernicious anemia. In this can be seen, but it is rare to find in pernicious anemia. Now in this pic you can see the oval jolly bodies which I circled. These are purple on Romanowski stain, so please remember this. Now our second body is Pappenheimer body. Pappenheimer body is peripheral basophilic inclusions in the RBCs, but the differentiating feature between oval jolly body and Pappenheimer body is that these are smaller than oval jolly body. So Pappenheimer are smaller than oval jolly body. In this pic you can see there is a peripheral basophilic inclusions in these cells, which is as you can get an idea that it is smaller than the oval jolly body. Now these are composed of hemosiderin. These bodies are composed of hemosiderin and can be found in sideroblastic erythropoiesis and hyposplenism cases. Now the stain used for these are pearl stain, as they contain hemosiderin, so we will use pearl stain, and this stain color as blue color. Oval jolly body stain as purple color on Romanowski, and Pappenheimer stain as blue color on pearl stain. So guys, let's and this is enough for today, and we will continue our series in the next video. So till then, please like, share and subscribe. I hope you are enjoying my videos by watching and understanding all the important topics. So please, for your support, I need you to subscribe my channel and press the bell icon. And thank you for watching my video. So let's meet again in the next video. Thank you.